Hello everyone, my name is Angie Sanhimi from 12B Group. Today we will discuss about randomized control trial or randomization in clinical test. In this video, we will talk about what is randomization, how are subjects randomized, what are different types of randomization, and why a process called randomization makes this trial the most reliable way of testing treatments. What are randomized control trials? It helps in treating, preventing, detecting disease and health conditions. Steps in randomized control trials: designing a protocol, selecting reference and experimental population, randomization, intervention, follow-up, outcome analysis, and publishing. Inclusion and exclusion criteria are the conditions that must be met in order to participate in a clinical trials. Age, gender, the type and stages of a disease, treatment history, any other medical conditions. Who should randomize? We need to separate the person who generates allocation from those who assess eligibility. Third-party scheme, telephone randomization service, pharmacy randomization, web-based service, envelopes, serial number, sealed and opaque. This trial tests many types of treatments such as medication, medical device, surgical techniques, physical therapy, and behavior therapy. We may compare new and old medicine or currently available treatment for a particular disease to figure out which one works best. Or a researcher find a new antibiotics and give it to a group of people and separate into two groups, give new antibiotics to a treatment group and standard antibiotic to a control group, and compare the result. Or they can give a placebo. It is a dummy drug with no active ingredients. What are different types of randomization? Types of randomization are simple randomizations, unequal allocation randomization, stratified randomization, block randomization. Simple randomization is based on a single sequence of random assignment. Basic method of simple randomization is flipping a coin in computer-generated sequence. 15 numbers of subjects are divided into two groups of equal number, treatment groups and control groups. Block randomization is for smaller studies. Participants are randomly assigned to treatment groups in small blocks of 4 or 6 participants. Ensure that they are easily sized groups meet the assumptions of equipoise. Stratified randomization, randomly assigning participants to treatment groups based on predefined and objective characteristics, example, age or gender. Each stratum is randomly allocated to treatment groups. Unequal allocation randomizations means randomly assigning participants into disproportionate size groups. The acceptable ratio for unequal allocations are 2:1, 3:1, and 4:1 controls to cases. This method is useful for studying rare outcome, adverse events, or emerging therapies. However, equipoise is not possible. Trials monitoring must be monitored by institutional review boards, IRBs, for ethics and safety. Trials should have an independent monitoring system to periodically review data and ensure participant safety. Data Monitoring Committee (DMC) or Data Safety and Monitoring Board should have the authority to terminate or change the trial procedures. Trials should report all adverse events, especially serious adverse events and unexpected events. Let's talk about the advantages and disadvantages of RCTs. In advantages, it is unbiased distributions of co-founders, blinding more likely, randomization facilitates statistical analysis. And disadvantages are expensive in time and money, volunteer bias, ethically problematic at times. The trial should ideally reflect real-world conditions. True groups that are comparable are free from bias. RCT is gold standard in terms of identifying casualties. Need to weigh theoretical advantage against practicalities. Randomization methods. Unpredictable, balanced, and simple. Thank you for listening.